Good evening, everybody. We're going to go ahead and get started. I want to welcome you to another installment of the WildCare Virtual Happy Hour. I hope you all are enjoying a cool refreshment or perhaps a sponsored cocktail for this evening, the Ninja Turtle, or perhaps an STL Zoo Bier Kölsch ale. So I'm going to go ahead and get it started. Before I do, I want to kind of go through a run of show tonight and let you know. So we have a panelist of 4 different panelists for this evening. They're going to be talking about turtle conservation. Before I get started with introductions, if there are any questions that you all have in terms of the presentation for this evening, please feel free to enter your question into the Q&A chat box that's located on the bottom of your screen. We will try the best that we can to get through all of the questions this evening, and we'll try and get everybody out of here on time. So I'm going to go ahead and introduce our panelists for this evening. I'm going to start with Dr. Sharon Dean. She's the director of the St. Louis Zoo Institute for Conservation Medicine. Our next speaker is Jamie Palmer. She is a technician with the St. Louis Zoo Institution for Conservation Medicine. And then our next panelist is Kathleen, and I apologize, Kathleen, if I get this wrong, but it's Kathleen Abucubical, something like that. I apologize for completely butchering your last name, but if you have a chance to say that during your presentation, that would be fantastic. I apologize once again. And then we have our last panelist. We have Dr. Marisbrin White, who is a research fellow for the St. Louis Zoo Institute for Conservation Medicine. So I'm going to go ahead and turn it all over to you. Once again, if you have any questions, please feel free to put them in the Q&A chat box, and we'll get through them as soon as we can. Thank you all very much.
Great, thanks, Mike, and good evening, everyone. We, as the ICM, really appreciate spending a little bit of time with you and talking about one of our favorite topics, turtles. So I get to kick us off for a few minutes just talking about why turtles make us happy, why we care about turtles, and hopefully let the others go into more detail about the work we're doing. Next. So as Mike said, I'm starting just Turtles Rock. I'm going to give us some information just about where turtles fit on our planet. Kathleen Abucubical will be speaking second, talking about one of our programs in Galapagos. Maris is going to introduce us to the work we're doing in Madagascar. Jamie ends us up talking about our local turtle conservation work. And then hopefully, if we do this right, we'll still have plenty of time for a Q&A and just to talk about turtles. And Carrie is going to help us moderate that. Next. So first of all, I just want to say we are the department, the Institute for Conservation Medicine. And if you're not familiar with a One Health approach, everything we do takes this holistic approach for wildlife conservation, public health, and sustainable ecosystems to ensure healthy animals and healthy people. So if you're interested in any of our work, please visit our website. We have a fairly robust, many web pages on the St. Louis Zoo website, and you can learn about all of our other projects. But as you're going to see tonight, we are a little bit turtle mad. So next. So really, myself, I kind of started being introduced to sea turtles doing a residency in Florida. I've worked with sea turtles across the globe. We won't spend time talking about sea turtles tonight, but that's really where my love of turtles began. Working in Galapagos next. As you see, Jamie's going to talk about some of the pond monsters that we've been working with recently. And then next, as many of you know, over the last 8 years or so, we've been chasing these iconic little box turtle creatures you can see on the one slide here with Maris. So we are fairly crazy about turtles, and there's a good reason for that. So next. So just a few numbers. You know, there are 356 species of turtles today. This picture I show here, you can upload that PDF off of Google right now if you want to. Unfortunately, half of those are threatened with extinction. There are also there are 7 species of sea turtles. Again, one of my favorites, I have to confess. And then lastly, next, here in Missouri, we have 17 species of turtles, which is unbelievable for a landlocked state. So really proud of how many turtle species we have right here in our state. So next. So, you know, what has a turtle ever done for you, right? I mean, they are cool little creatures, but they also are really, really important in our ecosystems. So if you think about it, they do everything but fly, right? They live on the land, they live in our oceans, our rivers, and they really have an important role in ecosystem health. So they're ecosystem engineers, and they can serve as both predator and prey, and they can also be used as sentinels for environmental health. So they're doing a lot for you, whether you realize it or not. So really, really important. Next. And then turtles are important in culture and religion. So if the next, there's a great book out there, Men of the Mississippi. It is amazing if you haven't read it. It goes back 100 years. Next, I think you'll recognize these, Crush from Nemo, the Ninja Turtles. I mean, these species are important, right? And then lastly, next, we also know that they really play a big role in many different religions. In the Hindu religion, you know, the tortoise is, the world is really carried on the back of a tortoise. They have been over millennia, they have been really important in our religions and our culture. And then lastly, before Kathleen kicks us into some of our projects, next, the one kind of bummer and downer at our happy hours, turtles are in trouble, right? 356 species remaining, half of them endangered with extinction. And there's a lot of different reasons for that. The illegal and legal trade, land modifications, impacts of ocean and rivers, infectious diseases, predation. Next, and something that you might not be thinking about, light and noise pollution. This poor leatherback here, you can see the beach far away. Lights from the beach resort has come out into the savannah. We're moving it back. So lastly, next, for all these reasons, they are amazingly cool, they're amazingly important in the ecosystem, and they have a lot of challenges right now. And Kathleen is gonna talk about our Galapagos project in this next section. So thanks for letting me speak at the beginning here. You're on, Kathleen.
Right. Hi, everyone. My name is Kathleen Apakupakul, and I am a research associate for the ICM. I manage the molecular diagnostics lab for the ICM, and I'm going to talk about a trip I took to the Galapagos to conduct pathogen diagnostics in these Galapagos tortoises. Next, please. So as you know, Galapagos tortoises are amazing. They are the longest lived of all the vertebrates, averaging over 100 years old, and they're also the world's largest terrestrial reptiles. Next, please. And they're scattered throughout the Galapagos Island archipelago, where about 2 to 3 million years ago, they arrived from the mainland by floating on these strong ocean currents, and they colonized 9 or 10 of the islands, and they evolved into about 15 different species. But then in the 17 and 1800s, humans removed up to 200,000 tortoises for consumption. So now only 10% of the original population still remains, with just 11 species left. Next, please. And human impact continues to be a problem, not just for the tortoises, but for much of the wildlife on the Galapagos. So you can see in the graph in the upper left-hand corner that the population of permanent residents and the stream of tourists has been steadily increasing. So you have to think about what kind of impact all of this development and the influx of people will have on these fragile island ecosystems. And another threat to giant tortoises is the presence of non-native species introduced by humans. So there's predation on tortoise eggs and hatchlings, there's competition for food, and proximity to livestock on farms adjacent to wildlife habitat threatens to expose giant tortoises and other wildlife to things such as antimicrobial resistance and diseases. Next slide, please. So in 2009, Dr. Stephen Blake, who's currently with St. Louis University, started the Galapagos Tortoise Movement Ecology program, which looks at the ecology of tortoises by tracking their movements and behavior. And then in 2013, the ICM joined as a co-investigator, which tacked on a health component to this program. So with the inclusion of a health component to the project, each tagged tortoise would receive a visual physical exam. We could test fecal samples for parasites and stress levels, and we could analyze blood samples to help determine an individual's overall health. However, we hadn't been able to diagnose specific infections since there's no equipment or trained staff in Galapagos to do the testing. And at the time, we weren't allowed to export samples to do the testing due to CITES or import-export restrictions of endangered species biomaterials. Next slide, please. So in order to be able to conduct diagnoses of infectious pathogens, I made a trip to the Galapagos back in 2018 to help our ICM graduate student, Dr. Ainhoa Nieto, set up a lab. She's a veterinarian originally from Spain, and she's currently working on her PhD with the ICM in the Galapagos. And she also works on staff for the Charles Darwin Foundation. And for her PhD, she's conducting a study examining the effects of close living with humans on tortoise health. So she's asking, what is the current health status of Galapagos tortoises? And are human activities negatively impacting their health? And she's doing this by comparing data between tortoises that live close to humans versus those living farther away. So our aims for this trip were to first provide molecular diagnostic testing of diseases on collected tissue samples to complete the health assessments. We wanted to provide capacity building by training Ainhoa in these techniques so that she may be able to conduct the work independently and then in turn train local staff and students on how to do this stuff. And then lastly, we wanted to pilot an ICM mobile lab for use in future research trips at our other field sites when needed. Next slide, please. And then for the most part, I worked indoors inside a lab every day for the 2 weeks I was there. But for my daily commute, I rode a bike from my apartment in town to the research station down a largely pedestrian road through a national park. And along this commute, I saw beaches and I saw all kinds of wildlife. And instead of dealing with car traffic, I just had to watch out for the occasional marine iguana crossing the road. And the weather was awesome every day. And it was just a really pleasant commute, so you can't beat that. So the first thing we had to do when I first got there was to set up the lab. And so since we're conducting diagnostics, we really had to think about where to set things up to minimize the potential for cross-contamination of our samples. And with the equipment I brought, we were able to test for adenoviruses, herpes viruses, and mycoplasma. Next. So this is the general work stream. So first, swab samples are collected from the tortoises in the field. So we swab their eyes, their nose, their mouth, and their cloaca. And on this particular work trip, I trained Ainhoa on how to do DNA extraction, PCR testing, and gel electrophoresis to figure out if these tortoises are infected with pathogens. Next. So in this photo, she's doing a DNA extraction, and what that entails is isolating the DNA in a sample and separating it from all the other crap, all the proteins and lipids and cellular debris, as well as all the dirt and the muck that the tortoises get into. So what you're left with is not only DNA from the tortoise, but also DNA from whatever pathogens that individual might be harboring. And this might sound familiar to you if you've had to get a swab test for COVID up your nose. So that swab sample is also used for PCR testing to see if you're infected with coronavirus. Next. So once you've separated out the DNA, you can check what kind of DNA is in the sample. And we target a specific pathogen DNA sequence using PCR or polymerase chain reaction. And in a nutshell, PCR allows you to pick out a pathogen's DNA if it's in a tortoise sample. So there might be very few copies of that piece of DNA in your sample, which would be very hard for us to detect. However, with PCR, you can throw in some enzyme and building blocks into a tube with that sample, and you make it go through multiple cycles of temperature changes in a machine, what the ICM likes to call the magic box, which makes copies of this DNA so you can, in essence, double, then quadruple, and so on the number of copies of that targeted piece of DNA up to a level where we can see it. So we can't see 4 or 8 copies of DNA, but we can see 1 billion copies when we run it on a gel. And really, all we want to know is a tortoise infected with a pathogen or not? So in our case, if a tortoise is infected, we should be able to pick it up with a swab and then amplify the pathogen's DNA that was on the swab up to the level of detection. So next slide. So using PCR, Ainhoa was able to detect a novel adenovirus and a novel herpes virus that had never before been found in giant tortoises. So this is a picture of a gel from a PCR test that Ainhoa did, and it shows a band. It's that second one from the left, which is the DNA from a virus called an adenovirus that came from a tortoise sample. Next slide. So we've found so far that, in fact, proximity to humans and their environments are affecting tortoises, at least through the discovery of antimicrobial resistance genes in tortoises that had been done with some prior work that she had done. That short trip that I took, it was just a pilot study. But since then, I know it's been able to take all this stuff that she's learned and just run with it. And what's great is that she's been able to train other people on how to do this work, so that it frees up her time to get other things done. And two, it provides scientific capacity building by giving them a new skill set so that locals can be the ultimate caretakers of the Galapagos tortoises. And in the process, we found new viruses for the first time in these giant tortoises, and we're still continuing this work looking for pathogens in the remaining tortoise populations left in the Galapagos that we hope will help with their conservation. Next. So that was just a quick overview of just a small portion of our program on Galapagos tortoise conservation. And now I'm going to turn it over to the next speaker, Marisbrin White, and thank you very much.
Hey, I think I'm hoping people can hear me. Someone in my chat room, give me a thumbs up. So I am Marisbrin White. I'm a veterinarian and currently a research fellow working with the Institute for Conservation Medicine. And I wanted to tell you about one of our newer projects on a whole different island, working with radiated tortoises. Next slide, please. So radiated tortoises are endemic to Madagascar, so another one of our island biodiversity hotspots. It's the only place in the world where they're found, and they are, like so many other really amazing species on islands, are critically endangered. The main reasons right now that radiated tortoises are endangered are because of habitat loss, and more we're seeing illegal collection. And what illegal collection means is taking of animals for food and primarily for the pet trade. So what you are looking at in this picture is a confiscation of over 8,000 radiated tortoises in one confiscation. And all of these tortoises were in a home getting ready to be shipped out of the country into the Asian pet trade. Fortunately, they were found, but then you have these 8,000 tortoises that you have to figure out what to do with. And the organization that is currently responsible for these tortoises is called the Turtle Survival Alliance. They have over 24,000 radiated tortoises in their care in Madagascar. So figuring out what we call the confiscation to release pathway is a really key part of conserving this endangered species. Next slide, please. So what I did, my role in this was to go to the Tortoise Conservation Center, which is one of TSA's, and again, that's TSA, Turtle Survival Alliance, not the people you see at the airport. So the Tortoise Conservation Center is one of their centers in southern Madagascar, where they were preparing 1,000 of these tortoises for release back into the wild. And I would love to show you tons of pictures of the Conservation Center, but because they do take care of these endangered species, with poaching being a big problem, it's a lot of security, but this is basically what the environment looks like. It's called the Spiny Forest in southern Madagascar. It's very desert-like, but very close to the ocean. It's a very Dr. Seuss-y landscape. It's a very interesting place. Next slide, please. And this is just a really short video clip. What I'm hoping it will show you is the pens that the radiated tortoises are in. So this is one of my study tortoises, and these are all subadults, so they're about 1.5 feet long to about 1 foot long, and they're in these open pens with these really short walls. So they're as close to a native habitat as they're going to get before release. And so my job there was twofold. One was to do pre-release health assessments, which is sort of what we were just talking about, and then also to establish baseline health parameters for these populations. So I'll take that one by one. So first, a pre-release health assessment. So what are we doing when we do that? We're basically asking two questions. The first question is is this tortoise healthy enough to succeed when it's released in the wild? So if you have 24,000 tortoises, you want to make sure you pick your healthiest, strongest candidates for release so that they're most likely to contribute to those wild populations. So they need to get a good physical exam, basic blood work, some basic health screening, just like you do when you go to a doctor. And then the other really important question that we want to ask is, these tortoises have been all over the place with tortoises from all over Madagascar. Maybe they've been mixed in with other species. We don't necessarily know their history. So have they picked up pathogens or diseases that could be dangerous to wild tortoises if we release these tortoises back into the wild? Next slide, please. And so that was the pre-release health assessment part. So I'm going to talk about the baseline health parameter section. And so this is really trying to understand just what's normal for this species. So a really interesting thing about veterinary work and conservation work with endangered species is a lot of times we don't know what we should expect if they are healthy. So we wanted to take this opportunity to figure out what is normal in these species. Next slide, please. So this was a big collaborative project. You can imagine I did not examine 1,000 tortoises by myself. And again, we worked with TSA Madagascar, which is primarily Malagasy staff or native, who are native to Madagascar, native Madagascans. And the other partners that we had there with us were the Wildlife Conservation Society. And this is our team. I had to Photoshop somebody in because he wasn't there on that picture day. Next slide, please. And actually our workup is pretty similar to what they do with the Galapagos tortoises. So we draw blood. That's what you can see me doing there in that first picture. We take swabs from the mouth and the cloaca to look for the exact same pathogens that we're looking for in the Galapagos tortoises. And we take general morphometrics. We measure them in all directions to try and get a good sense of what their body condition is, because it's kind of hard to see fat on a tortoise. It's not impossible, but it's a little difficult. Next slide, please. Then I also, while I was there, had the opportunity to look at some of the free-ranging tortoises. So like I said, this is in native radiated tortoise habitat. So just outside the walls of the center is perfect habitat as well. So we went through that same physical exam, blood work, swabs, all of that workup on the wild tortoises so we can compare these rehabilitated tortoises and see are they similar to the wild ones? How are they different? How does that indicate health or poor health? And one of the big challenges with working with the wild tortoises, that very first picture is really what the wild tortoises do the minute you pick them up. They tuck in their arms and they are so strong and you just have to wait them out. Like a solid 20 minutes of me just sitting there waiting for that tortoise to pick its head out. Next slide, please. And then we head back to the lab. So we were not lucky enough to have Kathleen there. So we did not do any PCR on site, but we do, again, standard blood work, just like you get done when you go to the doctor. So you can see in the middle picture is Jane, a technician from our Department of Animal Health, and Tony, a veterinary student, doing white blood cell counts, looking at immune function. On the left is our blood chemistry analyzer. It was overheating, so it got pampered and got some ice packs and a fan. That was Dahlia, our tech from Wildlife Conservation Society, looking longingly at her fan because she donated it to the analyzer for the day. And then in order for us to actually do the PCR eventually, we put everything in that huge blue R2-D2 thing strapped into the backseat of a car, which is actually a liquid nitrogen tank. And that's how we actually keep stuff frozen in the field and ship it back. Next slide, please. So what we learned, while we were there, we ended up examining about 1,200 subadult tortoises. And we only found about 50 that we thought weren't good candidates for release, which says really good things about the husbandry and how they're taking care of those tortoises at this center. The ones that we did find unfit for release were mostly for soft shells. And that usually tells us something is off with their mineral balance and their nutrition. And then the other thing we found out is that along with everything else that global pandemics are bad for, they're bad for research. So we are still waiting for our PCR results. We are still waiting for some other things to come through. So hopefully I'll have a chance to talk to you again and let you know what else we find. And then that wraps up Radiated Tortoises, and I will pass it on to Jamie Palmer to talk about our aquatic turtles work.
Oh, thank you, Maris. Hi, everybody. I'm Jamie Palmer. I'm the technician for the ICM. I will talk to you today about our North Campus Aquatic Turtle Survey. So this is not a far off place. This is in North County in Spanish Lake at the zoo's new property. Next slide, please. Can we go to the next slide? I think there we go. I'm going to go back to the next one. But to give some background, the zoo acquired this huge property in Spanish Lake. And the ICM was tasked, because of some of the work we've done before with aquatic turtles in Forest Park a number of years back, we know aquatic turtles pretty well at this point. And so we were tasked to look at the water systems at this site and then and identify the number of species in the pond. Sorry, my kids are walking by. And get an idea of what's there. And so can you go back a slide, Emily, now, please? So from the zoo standpoint, we had kind of one goal: understand what species are there and in what habitats, which ponds are better. But in true ICM form, we decided we have to ask a few more questions. If we have these turtles in hand, let's really get some information from them. So our big questions are just that: what species are present at North Campus? And then from pond to pond, the ponds are very different. Some are more managed golf course ponds. All are probably sinkholes in origin. And so some of them are more sinkhole ponds still. They're not managed. The water is not pumped in or out. They're a little more natural. And so we wanted to see, do we see differences between? And then how healthy are they, right? So this gives us a lot of idea of the habitat, how healthy the habitats and the environment is. So we want to understand things like infectious disease. In this site, we want to understand toxin exposure. So the aquatic turtles can be good indicators of ecosystem health, like Dr. Dean was saying early on. And so they can bioaccumulate toxins and heavy metals. And so it can help us identify that. Baseline health parameters. So we just want to do full health evaluations on these guys. So the nice thing about COVID, it might be the only nice thing about this pandemic, is that my whole team was here with us. So a lot of times we're in different places, and the ICM team has really been able to kind of, for a full field season, work together on a project, which has been nice. Next slide, please. There we go. Sorry about the mix up. So how do we do this? We go from pond to pond. We spend about 3 trap days at each pond. We set nets one day. The next day we pull the nets out of the water. We see what we get. And so from there, we process each turtle individually. So we do blood samples. We'll take blood. We also take swabs from their mouth and their cloaca. And then we do a lot of basic morphometric measurements. We check weight. And then we give them a full health evaluation, so a visual exam. We will also notch each turtle. So around the edge of their shells, they're called marginal scutes. We file in a 3-letter code into that scoot, and that helps us identify mark recapture. So we can tell you, if this individual, we had caught it before. Next slide. So once we take all these samples and put all the turtles back where we found them, we bring the samples back into the lab. We have a field lab at North Campus. It was simpler than bringing them back to the zoo. And we were able to do all of our lab work on site. So this is that baseline health stuff I was talking about. So we can do basic blood counts, we can do chemistry profiles in the lab, and then we separate out samples and bank them for other things. So for that infectious disease, that toxin screening and that type of thing. And so we were lucky this summer too to have some students, which has been hard for a lot of people and a lot of students not having internships. So we were able to train the next generation One Health practitioners and do it on a local level. So this project has been really nice for us during this time, and I'm pretty sure it's kept all of our sanity, being able to be out in the woods every day. Next slide. And so real quick, just by the numbers, at North Campus, there's about 10 ponds that we think have turtles. There's a few more that might have some, but they don't always have standing water, so they may or may not. 8 is the number of ponds we've sampled to date. So we've gotten pretty far. We're really close to the end. And we've identified 6 different aquatic turtle species at North Campus. So we're very proud of this number. This is a lot. So 320 is the number of turtles that we've caught. So these are the turtles we've had our hands on. Of the 320, 172 are the turtles with health evaluations. So some of these turtles are recaptures, but we have been able to perform health evaluations on 172 turtles at North Campus across 8 ponds so far. And then 7 is the number of waders that we have filled with water by either falling over in the pond or going too deep into the pond. And yeah, I think almost everybody on the team has had their waders full of water at one point or another, some water grosser than others too. So I don't know if the video is here, but we had a small video. Okay, thank you. Okay, good. So this is, I'm very partial to snapping turtles, but this is one of the large snapping turtles we have caught. And we call them dinosaurs, so we're always just fascinated by them. And so we had to take video of her walking back to the water. So yeah. And I think that's it. Once this is done, we might be open for questions.
So Jamie, I would say, just to start us off right off the bat, because this is one of the questions that I had. So for your love of snapping turtles, what is the largest snapping turtle that you've ever encountered? Because I know that they can get really large.
That's a good question. So before we started catching a lot of turtles and doing all the health stuff, we kind of piloted a net in the pond, and we were able to catch the biggest snapper I think I've ever held. And I think it was somewhere around 24 pounds, 23 pounds. And that was the picture at the beginning of my talk. That was that big male. His hands are huge. He was amazing. He also
that is massive.
Yeah.
Holy cow.
Yeah.
That was really cool. So we have a couple of questions to ask. I'm going to try and spread them out so that everybody gets a chance to participate in the Q&A section. So I'm going to start with Sharon. So this is maybe a juvenile question, but this is a question that may have come in. Can you tell us what the difference is between a turtle and a tortoise?
That's a great question. And I'm happy to get only the juvenile questions, by the way. It has been a long day for all of us. Not a juvenile question. So it's really semantics, right? So the tortoises are on land, right? And turtles can be in rivers, oceans, and on land. I think it's semantics. I think maybe the panel will disagree or chime in, but for me, it's semantics.
Okay, I gotcha. Well, good. So my next question I have is for Kathleen. And once again, I apologize for mispronouncing your last name. But this question is for you. So out of the work that you've been doing and working with tortoises, have you ever, or do you always tag them before they're released into the wild?
I actually don't do the tagging. It's the field people who do the tagging.
Mm-hmm.
So actually that might be a question for Jamie, because she's done all of the tagging. I know that all the box turtles in our study are not tagged, because that would get very expensive, first of all, and that would require a lot of manpower to track all of those turtles.
Okay.
Yeah.
Gotcha.
So Jamie, what do we do? Well, I was gonna say, what do we do instead of tagging? What we do instead of tagging them is we notch them. And so that way we can't go, so Jamie described that process, right? Of making the little notches in the outside of the shell.
Mhm.
And that means we can't go find them again. But if we do find them again, we know who they are. Like they're carrying an individual ID number, every turtle that we touch, so that when we find it again, we know we've already seen it. That way we can track its growth. We can do all sorts of things.
Gotcha. Okay.
Yeah.
So Maris, we're gonna stay with you. So the next question I have is for you. And you were talking about overall breeding and release into the wild. So I know that you talked about, I believe you said 1,000 tortoises that you all had reintroduced, I believe it was tortoises that you had released back into the wild. On average, is 1,000, is that normally around the average, or is it larger average?
Yeah. If that number, okay, so first we made sure that they're healthy for release. Releasing animals is a long, involved process. So this was one little step. The next step is they actually have to go to this pre-release enclosure and live there for 6 months so that they start to identify that as home. And then they'll open it up and start doing what's called a soft release, where they get food and water and support, but start to go out to the habitat. So these particular tortoises haven't been released yet.
Okay.
So I just didn't want to take credit for something I didn't do yet. But if 1,000 sounded like a lot, it's because it's a huge number. That's a very large number. And one of the super interesting things to me about radiated tortoises is when we think about endangered species, usually we're like, oh, this is the last 50 or something. Like that's sort of the popular idea. Radiated tortoises, we still have them in pretty big numbers. They have really good genes. They're a pretty robust population, but they're declining so fast that they're really dramatically threatened. It's kind of like if you know the passenger pigeon story, where like they used to blacken the sky and then within decades they were gone. It's like that. So endangered species can look sort of different, and because there are so many radiated tortoises right now, it makes sense to release 1,000 of them at one time across a few different locations so that they've got good habitat and good food and all that kind of stuff.
Okay, gotcha.
Yeah.
So the next question I have, this one's for Jamie, because you were talking a lot about the work up in North County. So my question is, how hard is it to actually work with turtles? Is the challenge, what are some of the challenges? Is it the challenge of actually wild, of going into the water and finding them and catching them? What are some of the challenges that are associated with that?
So it's a good question. I mean, there's a lot, it depends. You know, if you're with the giant tortoises, it's hard on its own level. Oftentimes for us at North Campus, at least, the ponds are, sorry, it's really loud on my end. The ponds are very different. Some of them we have to actually machete our way into them because they're in the woods. They're these sinkhole ponds. And so working with the turtles themselves, though, is the smaller ones are a little easier. The red-eared sliders are really mean animals. They want to bite you, understandably so. I would too. I think our boss would say that the snappers are the hardest. They're my absolute favorite, but they're snappy, right? And so their thing is to jump at you. And so we move with them kind of quickly to minimize stress on the animal, but we do it in a very unique way to try and like keep our fingers safe and our whole hands and that kind of thing. So the snappers are probably at North Campus the hardest individuals to work with, because they're you have to be on point the whole time with them, and the other guys, they're a little sneakier about when they would try and bite you. So yeah.
Okay.
And then getting in the water is always a tricky thing when you don't know what you're stepping on.
Yeah, for sure. That makes sense. So the next question I have from the Q&A chat box. So this is for Kathleen. So how long does it take to get to the Galapagos from here? And maybe more specifically, how many airplanes do you have to hop on in order to get there?
Okay, so it depends on where you're connecting. So when I went, we flew from St. Louis to Houston. So I think that's about a couple hours or so. And then from Houston to Quito, the capital of Ecuador. And I believe that was about 6 hours, a 6-hour flight. And then because of the timing of the flights, you basically have something like a good chunk of time before the flights for Galapagos take off. And so it's actually kind of worth it to get a hotel room to at least take a shower and get some rest. And then the flight from Quito to the Galapagos is about 2 hours. And, you know, that tells you about how many planes you have to take. But then once you land in the Galapagos, you have to get on a bus from the airport, and the bus takes you to a boat, and then you have to get on a small boat with all of your luggage across this little canal to get to one of the main islands. And so that part was really pretty challenging if you're carrying a lot of equipment.
Oh yeah, for sure. No, I can understand that. I bet you that takes quite a while. Okay, cool. So my next question I have, this one's for Maris. So in terms of the work that you've done in Madagascar, on a personal level, what is some of the, I would say, the most interesting thing that you've eaten within the culture that's there on Madagascar?
Oh, so I have to say, Madagascar is not the most exciting place for exciting food stories. Like I've eaten a lot of strange things a lot of places, and in Madagascar I ate beans and rice nonstop for however long I was there. And that's mostly, once in a while you get an egg, although what you do eat a lot of down south are prickly pear cactuses. And the tortoises love them. They're a totally invasive species. The French actually brought them in to make fences for their cattle. So there's like cactus fences all over the south, but they make these like beautiful prickly pears, and there's like a special technique for peeling them without getting the spines on there. And so, yeah, so I ate a lot of those, and they dye your whole mouth like bright purple, which is fun.
Cool. So I have another question for you, Maris, before we jump off to another person. Do you do any blood work on wild, and I know I'm going to get this one wrong as well, Astrochilis?
I do not. So Astrochilis. Well, so yeah. So Astrochilis is a radiata. So that's the genus of the radiated tortoise. And so there's more than one Astrochilis out there. But so we did do the blood work on both the ones in the Tortoise Conservation Center that were the confiscated ones that are going to be released, and did them on the wild ones. The wild ones, they're harder to find, and that wasn't the main purpose of the trip, so we only did it on a small number of 20. And it's sort of all different age ranges, because we'd find little guys who are only a couple of years old, up to, you know, the big like 35-pound tortoises, which, you know, is nothing in a Galapagos tortoise size, but for a radiated, it's big.
Gotcha. Cool. So Sharon, you're the next victim to my questions. So this one is a question that we had come in. Are there any diseases that humans can give turtles or tortoises?
Humans can give or get? Maris wants this one, I can tell. So is it, because really the one good thing about working with turtles and tortoises is that we're so different from them, right? Humans, it's not like working with non-human primates, let's say, where we share a lot of pathogens or diseases. The thing that we worry the most about, particularly here in the US, is Salmonella. People have probably heard about that. So Salmonella as a disease that turtles might give to us. There really isn't, we don't have a close relationship, so things spill over from us. Like, let's say if we were doing a project with non-human primates. But I know you're not going to ask me this question, but I do want to point out to our attendees, you know, listening to all these cool projects that people have presented here, and what we said at the beginning, you know, really it is interesting to think about how these turtles are playing a role. I think Jamie said it at one point, but are playing a role as sentinels for environmental health. Kathleen pointed out, like, we're looking at antibiotic resistance in the tortoises in Galapagos. That's a huge human health issue as well. Most people have probably heard of that. So really thinking about why it's important for the different projects we're doing, not only for wildlife conservation, but also for human health, and then for ecosystem health. So again, it's that one health concept. I know you didn't ask me that, but I just wanted to throw it in.
That added value.
There you go.
Maris, is there anything that you'd like to add?
No, I just thought it was a great question. So I was really excited. I was like, who did that? Props. That's what that was.
So Maris, I have one more question for you. So for your topic for tonight's discussion, they want to know, why are they called radiated?
Oh, I wish I just had a picture to show you, because if you Google them, because we can all do that, each one of their little scutes, so the scutes are each little part of the shell, like it's on all those little sections, and in each scoot, there's a little yellow spot in the middle, and then yellow rays coming out of it. So it's named after the pattern on its shell. It looks like it's got radiating stripes. That actually, unfortunately, is what makes them really desirable for the pet trade, because they're very like graphically beautiful tortoises. But that is where they get their name from.
So we already know this answer for Jamie, because she already talked about it during her presentation. So I'm going to ask this one to Kathleen. So out of all the turtles and tortoises, what would be your favorite?
Of the ones that we study, or in general?
Just in general.
So my favorite turtle is the diamondback terrapin, and that's because I grew up near the Chesapeake Bay. So I have this fondness for Chesapeake Bay wildlife. And when I was younger, I got a chance to see some baby diamondback terrapins, and I think they're just really cool because they live in brackish water, so like not quite salt water, but not quite fresh water, and they're just neat little animals. Sorry, other turtles.
I gotcha. So to keep it localized, I'm going to ask Jamie, I'm going to ask you next. So out of all the work, so since you're doing a lot of work up in North County, what is it that our viewers here tonight, what is something that they can be doing to help out in terms of conservation that they can do either at home or to educate the public of helping the overall turtle population within the St. Louis region?
That is a phenomenal question, and one where we often, like Dr. Dean just did, we offer the answer to that question, whether it's asked or not. So I've worked with a number of turtle species now, and I'm always shocked when people locally just don't really understand kind of that these guys are, this is we're in their space, right? This is their land. And so one thing we always say to them that's very simple, it's very to remember, is keep them wild. The way you can help turtles is by not taking them home as pets. They don't make great pets. They don't want to live in your basement. They want to live in whatever forested or water system you pull them from. And so we tell people all the time, you just leave them where they are. When they're crossing the road, you put them in the direction they're going, because they're going to turn around and go that direction anyway. They're going there for a reason. So help them out when they're there, but leave them be, because that's what the millions of years of evolution have primed them for, is that habitat. And so if you do nothing else, and I mean, talking about turtles to everyone you meet is also fun for all. And, people do love turtles. Dr. Dean always says it, that everybody has a turtle story and everybody loves turtles. And so that's the simplest message we can kind of give, is if you like them, if you love them, if you care about them, keep them wild and keep wildlands for them. And that'll go a long way, because they're in real trouble. And it's not just radiated tortoises. People are taking box turtles from Missouri for the pet trade and for Asian markets. And it's a local thing. It's not just across the ocean. And so, yeah.
Awesome. That was a great answer.
Thank you.
So now the next question I have, this is for all of you. So I hope you all are prepared, because I feel bad for Sharon, because I'm going to have you go first, and then this will maybe feed into the others. But throughout your career, in terms of all the work that you've done and the research and conservation work that you've done throughout your career, what is the one memory that has stuck with you throughout your entire career?
Oh, Maris, stop laughing. I don't think I can answer that. But Mike, I do want to share something with you. And that is the reason all of us are sitting here, right, and talking about turtle work, and the St. Louis Zoo Institute for Conservation Medicine has gotten so into turtles, is something that has stuck with me 9 years later. And when I first moved to St. Louis, I'm like Kathleen, I'm from the East Coast. I'm a sea turtle girl, as I told you. When my family moved here, somebody had found a box turtle that they rescued from Forest Park, and they asked me to check it out. It was living in her bathroom, and it was looking very sick to me, and she knew I was a veterinarian and she wanted me to check it out. And she said, oh, I rescued this box turtle from Forest Park, and my family and I just went on vacation to Miami, and we took the turtle with us and we put it on the beach so it would feel like it's at home. Mike, are you following this?
Yeah.
So again, my husband's from London. I'm from the East Coast. We went home that night and thought, oh my gosh, people here in Missouri thought a box turtle that was living in Forest Park was a sea turtle that needed to go home to Miami. And that's when we did what everyone does. We Googled and learned about the turtles of St. Louis. The three-toed box turtle is the Missouri state reptile. And we said, we need a box turtle project. And we started it in Forest Park. So that was just an example of here we are in this amazing state with 17 different species by this park where our zoo is located, and people thinking these box turtles are sea turtles. And we knew that we had a lot of just education to do. And that's why we've been doing hundreds and hundreds of kids on turtle safaris and just talking to people here in Missouri about our turtles and how they differ from, let's say, a diamondback that Kathleen might like, or a leatherback that lives in Florida. So that's the story I'm going to give.
Okay, now that was good. So I'm going to go with Maris next because you were laughing, and let's see if you can top that one.
I probably cannot top that. I'm not going to lie, but I was laughing because I cannot even begin to approach the question of like, what is the most impactful story that I could think of. But so I'm going to pick a sort of moment that happened to me several times in Madagascar, since that's the project I shared with you guys. And really, it's just so if you can remember the picture of the Spiny Forest, it's dry. It's called the Spiny Forest because everything has these like 1 to 3 inch spines on it. So it's not an easy place to walk around. And there's very few things there that are not reptiles and insects. The cicadas are super loud, like the cicadas here, but just millions more of them. And you're working all day and you're just super focused and you're pouring sweat because it's 100 and something outside and you're covered in dirt. And the tortoises just like keep peeing on you every time you pick them up. So you're covered in tortoise pee and red dirt and cicadas are screaming. And you hit this time of day where you're just exhausted and done. And then right about then, it just starts to cool down a little bit. And the cicadas kind of quiet down. And all of a sudden, this like group of ring-tailed lemurs would come running over to the lab clinic to see us every night, like right around 5:30. And you're just beat. And all of a sudden you realize where you are, like you're not in work anymore. You're like, I am in Madagascar. There are lemurs watching me do this. I don't care how tired and how sweaty and how many cuts I have and how much tortoise pee is on me. Like I am lucky to be here. This is a special, special place in the world. So that's my story.
Okay. That wasn't bad.
Yeah.
Kathleen, I'm going to go to you next.
Yeah, those are hard to top. So I was kind of like going through my brain about like, oh, what's like the most memorable experience I've had? And it's really hard to pick one. And I kind of wanted to like tell you an invertebrate story because I love invertebrates and they're kind of the underdogs of the animal world. But I'm going to copy Maris a little bit and basically tell you that for my graduate work, I spent a field season in Kenya, and I was studying a species of bird called superb starlings. And they're beautiful, beautiful birds. And I worked at this place called Impala Research Center, which is north of Nairobi. And so it took a while to get there, and I met up with the research associate affiliated with our research lab. And basically my field work consisted of going into this janky little Jeep out into the field And we were doing research on 9 different populations of these birds. And so we had to basically catch them and take samples, kind of like what we do with the turtles, but birds. And I remember like our really fancy, sophisticated bird trap was basically kind of like what you see in the cartoons with like the net and the stick that's propping the net up tied to the string. And then there's like a long string that you kind of like hide behind a bush or something. And you just put down some bait. And then when you see a bird in the trap, you just pull the string and then you trap the bird and you can take all the samples, and thinking like, wow, this is super high tech, science, but, it worked, right? And because you work in the field, there's things you have to do to the samples that you collect, and you have to be able to process them, a lot of times like right then and there because of sort of stability issues or whatnot. And so for some of the samples we collected, we had to centrifuge or spin them down real fast, like blood samples and things like that. And so in order to do that, we basically had a field centrifuge that we had to connect to a car battery. And, if you could see this image of this really kind of broken down red Jeep with the hood up, and like you could see the engine, and there was like things connected to the engine, connected to this field equipment, and we're putting our samples in there. And, it's amazing what people can do in the field with just sort of like the limited resources that they have. And so it gave me an appreciation for, sort of the innovation that people come up with, if they only have so many things to work with. But, like we made it happen. And I would also say like in Kenya, kind of like Maris's experience in Madagascar, it was like, seriously, like someone drops you in the middle of the Lion King. Like you go there, you drive through, and it's just like the animals don't really care that you're there, right? So it's like, oh, there's an elephant on the side of the road, as if it was like, a cow in the United States. There's like, oh, there's a giraffe, and, all these animals were just all over the place. So that for that first week I was there, I was just taking pictures like crazy. It was just amazing. Like all this wildlife was just on top of me. And my friend, my colleague who is Kenyan, my friend Wilson, like I was trying to teach him some American lingo and memes and stuff. And so I was giving him this version of like the whole, why did the chicken cross the road joke, right? Except I was saying like, why did the elephant cross the road? Because that's what they have there. And he had never heard the joke before. And I told him like, to get to the other side. And he was like, paused. And he was just like, that's it. And I'm like, yeah. And he's like, I like it. I like that joke. So I don't know. I think about that as one of my most memorable experiences, like doing science and conservation in the field.
Kathleen, our modern MacGyver. So Jamie, I've given you some time to think it over. So I'm going to finish this question off with you. I'm going to turn it over to you now.
It's just a memorable, I forgot the question. I got wrapped up in the answers.
So out of all of the work that you've done, whether that be through research or conservation, what is the one memory that has stuck with you throughout your career?
Okay. So it doesn't have to be turtles then, I guess. So I've worked with Dr. Dean for a number of years now. I'm not a vet. I'm not a vet tech necessarily. All of my skills have been learned on the job. And I'm a former zookeeper. And so we got an opportunity to work in Cuba on Cuban Crocs, our team. And my boss is like, you're going to lead this and we're going to send you down there and you're going to be the health expert. And I'm like, oh, help us. Because I'm not a vet, but I've learned from really good ones. And so we get down there and it's me, a couple keepers from different zoos, WCS, Smithsonian, our herp curator, Lauren Augustine at the zoo, who's a huge fan of Cuban crocodiles, and who invited us on the project. And we went to do the first ever health evaluations of Cuban crocs at a farm. They're farmed crocs in the Zapata swamp where they're from. So critically endangered crocodiles. I thought there's no way I'm gonna love a crocodile like I do a turtle. Sure enough, I do. I also love the people that work on these animals. And so I was in a room full of herpetologists, could understand nothing they're saying, but I was the one they were there for, because I'm like, we're going to bleed the Crocs. It's all going to work. And the whole time I had like my boss in my head. She's like, you're fine. You're going to be great. It's great. Because she's such a trusting boss. And the whole time I'm just like this nervous wreck, like, oh God, they're all gonna see through me and see that I'm not a vet, because I'm not a vet and I didn't pretend to be one. But the first time I got a blood sample from a croc, I held it up and I was like, Lauren, I got it. And I did good. And she just looked at me like, stop talking. Now do what you do. But it was so, I was just like, I can't do this. And we were able to teach a whole room of people why health matters, and why just breeding the Crocs and breeding and then throwing them in the wild isn't a holistic way of doing it. And we had good conversation and I learned from a lot of people. And it was the first time I was able to kind of lead a project, and it was due to my training with our team, and I worked for some of the best. And so it was really, really nice. But then we also got to, so they have this hatchery, this crocodile room of babies. And I never in my life thought I'd hold a baby Cuban crocodile. And then he's like, come on in. And we figured it'd be like this very scientific room. And we opened the door and crocs are like running everywhere. And so the scientist in me is like, you don't know what clutch they were in. You don't know anything. And instead, they're just like, do you want to hold this? I'm like, I want to hold one. And so I got to hold a baby croc, and it was just a really memorable trip, because like all of the stuff we do, and I mean, I could chase a box turtle. If that was the only species I worked with, I could do it the rest of my career and be content, I think. But it was just a really good experience, and I worked with really great people. And yeah, that was a little rambly, but.
Oh, that was really, really good. So we're right at 6:31, but we do have one more question. And I think that this is an important one, especially with the 4 panelists that we have this evening. So I'm going to throw it out there. Either one or maybe 2 of you, maybe perhaps to answer this question. I'll leave it to you all to decide who that is. Within maybe a 30-second answer, maybe a minute answer. The question is, it starts with a statement. It's so great to see so many women scientists. Did you always want to study turtles, and have you overcome any obstacles while doing so?
You're only going to give us 30 seconds to answer that? I mean, give me a break.
In the interest of time, it's a very good question. And all right, I'll tell you what, I'll give you a couple minutes, Sharon, if that is what you need to answer.
I'm just kidding. But I'm happy to do a quick answer for I don't want to take it away from others, but turtles are amazing. I think I might know who asked this question, but maybe not. Turtles are amazing. I've always been fascinated with them. Just like Kathleen said, I grew up on the East Coast. I think turtles are an amazing species for young scientists that are getting into it. They are important ones to study, and they are very diverse to work with. I think the other question was sort of, have you hit some, some challenges to get in this field? I do think, there are a lot of obstacles out there for a lot of people in terms of getting into conservation positions, to having really good careers like all 4 of us on this panel have right now. And we all have different skill sets. We did different education paths. I think the challenges are still there for many of us, but I think there is a lot of positive things happening now in the turtle world. And that's why you see a panel of 4 women right now that are doing these, I think, amazing things with turtles. 40 seconds, I did it.
Well said, well said. Is there anybody else that'd like to share anything in regards to that question?
I just want to say we do have an amazing team, and it's the reason I come to work every day. It's not just the animals, it's our team. And it just is we'd take a guy if he made sense, but having an all-female team has been it's really nice. It is empowering. It's a really great thing.
Awesome. Well, thank you all so much for coming in and presenting this evening. And thank you all to our viewers for coming in and listening. It's definitely a very important topic and something that was very, very interesting to listen to. So if anybody wants to learn more about the WildCare Institute or the St. Louis Zoo, or even specifically around our conservation work around tortoises and turtles, visit our website at stlzoo.org, or you can reach out to any of these fine women around any of the questions that you have. Thank you again for coming this evening, and we hope to hear from you soon. Thank you very much.
Thanks, Mike. Good night.